Hey guys, welcome to our 2024 January series A-Time Notes lectures. So today we're going to be covering kind of like a brief overview of 1-2 bio. I know there's like an insane amount of content in here and I'm not going to go over everything in like a huge amount of detail, and I might even gloss over a few slides, but it's all kind of here so that you guys have like kind of like a set of notes that you can kind of refer to later on in the year. So I will try and focus my attention mostly to Unit 1 stuff, or things which I think are probably more high yield or more important, especially for Unit 3, 4 as well. So yeah, let's get started. So firstly, I should probably introduce myself. My name is Angelica and I'm a med student at Monash. I've been working with A-Time Notes it's my 5th year now actually. So ever since I graduated high school, I've been tutoring bio, PE and English with A-Time Notes and ShootSmart. So yeah, I love it. It helped me out a lot in high school too. So I think it's kind of like a nice little circle that I'm working with them, helping high school students now with A-Time Notes. So it's really good. Anyway, just a bit about A-Time Notes. So we were kind of established as a means to even out the VC playing field. So I know that VC is pretty intense. It's quite full on, and it can be kind of hard to find like a bunch of notes or resources all in one place. And I find that A-Time Notes does that really well. So it's all completely free. You can make an account. I made an account. I had like a fake name or anonymous name or something like that, so no one would see who I was. But I did that in high school, and I found it really useful for me. So there are tons of study notes which you can download, which are all written by students who've scored really highly in their respective subjects. And they're all kind of distilled in a way that kind of appeals to like VCE students and kind of recognizes areas where students particularly don't excel at, or perhaps areas which may need extra like mnemonics and things. So that's really useful. We've got lectures like this one. So special thank you to our sponsors. And also, if you are looking for any other lectures or any other subjects, feel free to like join them or like have a look on the timetable and see what else is out there, because we have got quite a few on this week. So yeah, just joining them, even just getting the notes from them if you can attend, could be really helpful to your learning. So I recommend. I also highly recommend the discussions. So this is what I used the most when I was in high school. I would like spam the bio forums because I found them really useful. So I would like do an exam, and anytime I came across a question that I was a bit unsure about, or any question which I thought like maybe I got wrong or needed a bit more help with, I would just put them in the forums. And at first I was really nervous to do it because everyone answering was like a 50 bio person, or someone doing medicine, or someone with like a 45 study score, and I'm like, I'm doing 3-4 bio and I feel like I don't know what I'm doing. But yeah, everyone was really helpful, and I get replies really quickly, like sometimes within like 30 seconds, which is crazy, but also super helpful to your learning. So there's like one thing you take away from this. Well, hopefully you take away a million things from this, but hopefully you also make an account. Like it's completely free. Like I don't really see any real caveats to doing it. I just found it super helpful, and the forums especially was really, really like instrumental in my learning in year 12 and year 11. So the videos are also really great. Lots of like bite-sized study tips and techniques, and like little explanations of things. And then the newsletters, just to keep you in the loop. I always get so worried this is not recording. It is recording. Okay, is it recording? Yes. Okay, sorry guys. And we've also got the ATAR calculator. And so this is something that I wasted too much time on in high school, I think. But it's kind of fun to see how you're tracking and see if perhaps you will attain your dream ATAR if you can predict your study scores. And we've also got articles, and these are all written by students who also scored really highly as well, or even students who are doing like uni subjects and things, which you might be interested in. And heaps more on our website. So check it out. It's completely free. You can even like have a look on the website, see the forums and things without even making an account. But if you do want to ask a question, you'll have to make an account, which you don't have to pay for it. It's not like a hidden credit card fee or anything. It's like free. So definitely recommend. And we've got tons more resources. So yeah, do check it out. Okay. So welcome to our Bio 1 and 2 lecture. It is going to be quite a lot of talking. It is a 2-hour lecture, so get a drink, settle in, get your notes out, because it will be quite a lot. So I like established before that I'm going to be covering a lot of content. There is quite a bit which I won't spend a lot of time on. I really just want to make sure that you guys have kind of a foundation which you can build upon. because I remember doing bio. So I actually did bio when I was in year 10, and I was kind of like overwhelmed by a lot of content. I was like, I've never done this before. I don't even know what diffusion is. And so it felt like such a big jump from like year 9 straight into year 11 bio. So what I want to do here is kind of help you guys like get prepared or feel like you're not coming across something completely alien to you when you actually start it during the semester. Hopefully it does help you out a bit and it does make sense. I actually find that the bio gets more interesting as like you progress along the year, and like I really enjoyed Unit 1 2 bio, but I like loved Unit 3 4 bio. I thought it was amazing. And then I got to uni and did even more of it, and I was like, wow, I love biology so much. So it can be a bit difficult at times and quite content heavy, but stick with it if you want to, because it is really well worth it. So yeah, welcome to our VCE Bio 1 2 lecture. My name is Angelica. Oh, my laptop is frozen. Why is it frozen? So we're going to be covering quite a lot of content, and put your messages into the live chat throughout, and I will message back and answer. Make sure they are appropriate because someone will be moderating them. But yeah, if you've got any questions or you want me to stop and explain, I won't stop because this is like a pre-recorded lecture, but I will be answering everything live in the chat. So yeah, please put any questions you have. So as you can see, we'll be looking at cells, organelles, plasma membrane, cell cycle, plant vascular systems, animal systems, digestive, endocrine, excretory, and homeostasis. And I'll do the best I can to answer any questions. So yeah, even things about like biomed or uni or medicine, stuff like that. So yeah. So as I mentioned before, my name is Angelica. I'm a writer and a medical student, so I'm actually like a journalist for a few magazines. I graduated high school in 2019 with a 49 in biology, a 47 in English, 40p, and an ATAR of 97.25. So I actually did a Bachelor of Biomedical Science at Monash, which I really loved. I actually have quite a few students going into that now, and I honestly really loved biomed, and I don't regret doing biomed before doing medicine. Like I can't imagine jumping straight into medicine anymore. I just like think, wow, I'm so grateful for my time at biomed. So if you're thinking about pursuing biomed or even med, let me know. I'm now doing medicine, so I just did my first year med. I'm moving into second year. It's really like third year, post-grad year. So I'm going to be doing clinical schools all this year and onwards my degree. So I'm loving that too. I have 2 cats and a dog, and whenever people ask for a fun fact, I don't know what a fun fact is, but during COVID, I ran a virtual half marathon. So it was like a tracker on my phone and stuff. But it sounds so much more exciting when you say a virtual half marathon, like I've been running through like an AI landscape or something. Anyways, yeah, so that's just a little bit about me. Hopefully you enjoy this lecture. As I mentioned, I've been teaching bio for quite a long time now, and I am a complete nerd for it. So yeah, let me know if you find this useful. Okay, we're going to jump straight into it. So as I mentioned, I didn't have much experience in like bio jumping into it. Like I feel like when you're in high school and you're starting off, you do these like random experiments and things, but it feels like there's not enough context for you to structure what's going on. That's how I felt anyway. So when I jumped straight into like year 11 bio, I was like, I don't even know what a prokaryote is. So there was quite like a big learning curve, and like I'm being really stressed because I was like, I don't understand osmosis or diffusion, and I would like memorize words like concentration gradient and stuff. But it took a long time for me to kind of find my feet, and I think it's just because I didn't really have much background. So what I'm trying to do today is give you guys that kind of foundation so you can build upon it and hopefully feel comfortable with approaching these topics when you actually get to it in class. So we've got 2 main cell types, prokaryotic and eukaryotic. I kind of remember these as like pro, meaning like old, and eu, meaning like new, because it rhymes with new. So like if someone's a coach, they're typically like if you're playing golf, for instance, and you've got a coach teaching you and they're a pro golfer, they're typically going to be a lot older than you. So I always think of pro as being like an old thing. Like an old structure. Whereas you rhymes with new, so I think of it as being a newer one. And so what I mean is like pro is like the more ancient, more rudimentary, less developed one, whereas eukaryotes are like the newer and improved model, sort of, like in very basic terms. So we're going to kind of go through what they are. So they are the 2 main types of like cells. So we've got prokaryotic and eukaryotic cell types. Okay, so like biology, you're definitely going to be studying cells, and the two main types that we look at are prokaryotic and eukaryotic cells. And each of these cells have a different kind of like characteristic or different structures. And so with prokaryotes, they don't have a membrane-bound nucleus. So even the word nucleus might be new to you, because it was certainly very new to me. And I was like, I vaguely know what that is, but not really. So nucleus is like, they call it the headquarters of the cell. It's where all the genetic material is stored. So in terms of prokaryotes, they have this like nucleus-looking structure, but it's not exactly nucleus. It's kind of like the DNA free floating inside that cell. They don't have any membrane-bound organelles, meaning that these organelles they've got inside them, they're not wrapped up in a membrane. They're not really capable of true multicellularity. So prokaryotes are typically like organisms which are just one single cell, okay? Like a bacteria, maybe just one single cell is a prokaryote. And they typically reproduce via binary fission, which means they kind of straight up just clone their entire being into another clone of an entire being. And they just keep doing that. So that's binary fission, and that's how they reproduce. There's no like gestation or like reproductive cycle or anything like that. The whole thing just reproduces itself. In terms of eukaryotes, they have a membrane-bound nucleus, so that like headquarters of the cell is contained within a membrane. They also have organelles, which means they're kind of like wrapped up, kind of like when you wrap up presents in Christmas wrapping. They've got like a membrane which is wrapping them up. And they reproduce via mitosis and meiosis. So I'm not sure if you guys have heard of this word yet, mitosis or meiosis, but they're kind of methods of replicating cells, and they're a bit more complex, I think. I wouldn't know if they are more complex than binary fission. They both seem quite full on to me, but it's kind of a method of cloning, but more advanced, I would say, in very simplistic terms. But I think we will go through that later on. So I have this slide here. I know it's completely blank, but you guys get this slide deck as like a PDF afterward. So if you have some time, it'd be really great if you could just go through and try and label all the organelles. So I might give you a bit of a clue here. Like we've got the nucleus there, and we've got like smooth and rough endoplasmic reticulum, we've got Golgi, we've got mitochondria. But yeah, I think just being able to label all the organelles in this cell, this eukaryotic cell, is a really good practice, and also being able to define what each of those organelles do or their roles. So I've actually got a big summary table here. These definitions are very textbook-like definitions, which is fine, and is what you need for the exam or your SACs and things. But I find it easy to remember stuff if there's a story attached to it. So I like to make up like a little story in my head or a very visual picture. So you might notice that a lot of my like mnemonics are very visual. That's just how I remember stuff. Anyway, so the nucleus, people say, is like the headquarters of the cell. It's like where the genetic material lives. It's actually a double membrane. It's like wrapping something up. Like in Pass the Pass, we've got several layers. This has 2 layers of like wrapping around it. And it's porous. What this means is that it has little holes in it which allow things to kind of slip through and enter the rest of the cell. We've got ribosomes. Ribosomes are the site of translation, which is something we haven't come across yet. Probably, hopefully, maybe. I don't know if I say hopefully, but something you may not have come across yet. But translation is a part of the synthesis of proteins. So I think of ribosomes as like factories making proteins. And like when I try and remember things, I often draw like a little picture. Like I might draw like a little factory, like with puffs of smoke and stuff and write proteins. The protein factory. It's actually made of ribosomal RNA. It's not membrane bound. This is something I find crazy. So we have DNA, we've also got RNA. So RNA is like a single strand of genetic material, whereas DNA is like a double strand. But ribosomes are literally made of like strands of genetic material coiled up around proteins, which I think is crazy. But yeah, we've got the rough endoplasmic reticulum and smooth endoplasmic reticulum. I know these are like really long words, but you can call them rough ER and smooth ER. So rough ER has ribosomes attached to it, so it's got a lot of little protein factories attached to it. And what it does is it actually transports and modifies proteins to package them up and get them ready. Well, not really package it, but sort of modifies them or makes improvements and things. Smooth ER doesn't have any ribosomes on it, but it synthesizes steroid molecules and like lipids and fatty type things. You've got the Golgi apparatus, which modifies and packages proteins into vesicles. So I think of the Golgi apparatus or the Golgi body as like the post office of the cell. Like it packs stuff into like little parcels and gets them ready. Yeah, and we've got lysosomes. Lysosomes are like the recycling center of the cell. They break down unwanted stuff and like get ready to like send it out and destroy it. So they're really good at recycling. Something else I also find really useful in bio is to think of like the origins of the word or the etymology of it. So to lyse something means to kind of destroy it in like I think Greek or Latin or something like that. So to lyse something means to destroy it. So lysosomes are like little cellular structures which destroy stuff. Mitochondria. This is the site of ATP production. I know people go, oh, mitochondria is the site of, what do people say? I'm sure someone will like message in the chat. Mitochondria. Oh, the powerhouse of the cell. People love to say it's the powerhouse of the cell. Please don't ever write that in your exam because VCA doesn't like that, and I don't even know what that means. The site of powerhouse of the cell. I don't know what that means. What I would like you guys to say instead is the site of aerobic respiration, which we haven't explored yet, but we will over the next few years in quite a lot of detail. So yeah, mitochondria is the site of aerobic respiration or ATP production, okay? Not the powerhouse of the cell. So please don't say powerhouse of the cell in any SACs or exams or anything. Finally, we've got chloroplast here, which is the site of photosynthesis. So our cells, like animal cells, we don't have chloroplast, but plant cells will. So we look back at this diagram, we've got like the Golgi, probably this one. We've got smooth ear, rough ear, studded with ribosomes, nucleus with nucleolus. We've got mitochondria. It's a bit hard to tell what everything else is, but try and have a go in your own time. Or even get a picture of a cell and which is fully labeled and hide the labels on it and then have a go. Cool. Okay, so that's our organelles kind of summarized. Another good tactic is to like draw a picture or like write out nucleus and draw like a, what would you draw the nucleus? It's like a parcel containing genetic material. Or I love drawing the Golgi. I draw like a little post box with like envelopes and stuff because it helps remember it as the like the post office of stuff, packaging parcels of proteins. So here's kind of like a close-up picture of the mitochondria, which is our site of aerobic respiration. So we can see it's got a very convoluted internal structure, lots of folds and cristae and things. So we have a close look at it. It is membrane bound. You can see there's an outer membrane and there's also an inner membrane. And so this inner membrane has got folds. So it's got a matrix and cristae, which are the two main components of the folds of this inner membrane. We've also got, like, there are these words, intercrystal space, peripheral space. I wouldn't really bother memorizing them. Mainly matrix, cristae, they're the main parts. You also want to note that you've got mitochondrial DNA. So you've got your own DNA and then your mitochondria also have DNA as well. And mitochondrial DNA is really cool in that you can actually trace the mitochondrial DNA up through like the maternal line all the way back to this ancestor we have called mitochondrial Eve. So she's like the original mother, which I think is so wild. We can do that. So yeah, mitochondrial DNA only really carries the maternal line. So that's something to note. We've also got some ribosomes in the mitochondria because we know ribosomes are the protein-making factories and mitochondria need lots of proteins to make energy. And we've got this thing called ATP synthase, which maybe you guys can see studded in the membrane there. And that ATP synthase is a special enzyme which actually makes ATP energy. So I also teach PE. So if you're doing PE, you'll learn lots about ATP this year. But you'll also learn it in bio anyway. So ATP is adenosine triphosphate, which is a molecule which is pretty much completely vital for the contractions of our muscles. It's like the form of energy that our body uses. So here is a nice picture of a chloroplast. So chloroplasts are only found in plants. They are a kind of structure which is utilized for photosynthesis. So there are quite a few different components here. So we've got grana, or granum. I say grana, which is a stack of thylakoids. So if you kind of imagine like matcha pancakes, a thylakoid is a single pancake, but a stack of them is called a grana, okay? So one individual matcha pancake is a thylakoid, a stack of them is a grana, okay? They're connected by these lamellae, I get the little laser pointer out, which like draws them close together. You've got this like fluid filling the space, and it's called stroma. So stroma is like the fluid-filled space. It's got quite a high pH in it, which is conducive for photosynthesis, I guess. You've got some DNA there. You've got some ribosomes, once again, protein factories. Got some lipids, some starch, etc. Bit of a complex-looking diagram here. We won't delve into it too much, but it is about like photosynthesis and producing ATP energy and assisting with like energy production and glucose production. Okay, so that's kind of a bit of a basic background to our like general cell structure and some particular organelles which are quite useful. Now we're going to go into like the biochemistry of it and cell structure and function. Okay, so hydrophilic and hydrophobic. These are quite big words. I, once again, really love etymology. I just find it so useful for science subjects. Like if you ever do anatomy, learning the etymology or like the basic origins of words is super useful because it really helps you to kind of break down what a particular structure may be. So hydrophilic, and hydrophobic. We know phobic means like scared, right? If I'm arachnophobic, I am scared of arachnids, like spiders. But if you're hydrophobic, this means you're scared of hydro. And what is hydro? Hydro is water. Like a hydro farm is like, or a hydro plant or like energy producing areas where they use like hydro or electrohydro technology. It's using water technology, right? So hydro is water, or hydrate, water, right? So hydrophobic molecules are scared of water, okay? They really hate water. They don't want to be near it. On the other hand, philic means to love. I know people talk about being like a chocophilic and they're like obsessed with chocolate because they love it so much. So philic, to love, water is hydro. So hydrophilic means to love water. So it's a water-loving molecule. What this means is they want to be with the water. They want to, be close to it. They can actually dissolve in water. So they've actually got the property which allows them to dissolve in water. They may also be called lipophobic or similarly sort of polar. So yeah, hydrophilic molecules are polar. They love water. They hate fats. Lipophobic. So lipo is fat hating. And yeah, proteins are an example. I don't know why that's connected. Hopefully you guys can still hear me. I don't know. I might just. And turn that off. Okay, good. I don't know why I did that, but I was a bit worried for a second. Anyway, so hydrophilic and then hydrophobic molecules hate water. They won't dissolve in water. You might hear the phrase like dissolves like. So proteins which love water can dissolve in water. Hydrophobic hates water, so it won't dissolve in water. And they'll actually interact with other hydrophobic molecules, which are lipophilic or fat-loving or non-polar, so oils and fats. And you can actually see this in action if you have like a glass of water and you pour some olive oil on top of it. So you'll notice the oil will float to the surface, right? And that's because it's hydrophobic. That oil hates the water. It doesn't want to dissolve in it. It won't dissolve in it. It will just like stay as far away as possible from it, and it will just sit on the surface. So you can see this. You're trying to mix it up, but eventually that oil will just settle on the surface. Okay, so you can see that happening. They're separating to water and oil. That oil will not mix with the water. Okay, so that would be a hydrophobic substance. It's scared of the water. It hates the water. Which is really interesting because our plasma membrane is actually made of a mix of hydrophobic and hydrophilic molecules. So I might actually spend a few minutes talking about what a plasma membrane is. So our cells, they all have membranes. So they've got a plasma membrane. So it's kind of like separating one cell from another. If you think of like a bubble, it's like the outer shell of the bubble. Okay, so it kind of separates one bubble from another. You've got bubbles joining each other. So it's kind of like that outer packaging layer, or like that layer of wrapping paper around the present, something like that. Okay. And it's often called or referred to as having a fluid mosaic model, which is quite a long phrase, and we can break that up. Fluid meaning it's constantly shifting and evolving and moving about. Very fluid. It's dynamic. It moves. It's not very static. It doesn't stay still. It kind of like swaps about and moves. And it's a mosaic because it's made of very many different pieces. Okay, so it's a fluid mosaic model. And mosaic because it's got all these different pieces. You've got these things here with the tails, which we'll look at in a second. We've got proteins, cholesterols, carbohydrates, sugars, things all over. So it's made of very many different molecules. And you can see it's like that outer shell or layer of a cell. So phospholipids. What is a phospholipid? So phospholipid actually makes up that fluid mosaic model, that membrane, plasma membrane. And we actually refer to the membrane as a double membrane bound structure. Double phospholipid layer or lipid bilayer. So it's double. It's not double membrane bound. I don't know why I said that. It's got double layers of phospholipids or 2 layers of phospholipids. So it's a bilayer, 2 layers. Bi meaning 2, layer meaning layer, I guess. So phospholipids are amphipathic, which is quite a big word. But this means they've got a hydrophilic head. Hydrophilic meaning water-loving head. So that head is that red thing. It's made of phosphate and glycerol. And 2 hydrophobic fatty acid tails. So those tails hate water. They want to be away from the water. So if we look back at our membrane again, we can see our phospholipid. We've got that head, which is hydrophilic, water-loving, and the tail, which is water-hating, which is why the tails pack in close like this. They want to angle so that the tails are close to each other with the heads at the ends. And like surrounding the heads will be water. So this should be like all water here, whereas this area probably doesn't have a lot of water because these things hate water. They want to be away from the water. Okay, whereas the heads love the water. Yeah. So I've been talking a lot today, so I need to stay hydrated. Extracellular, intracellular. So this is the intracellular part of that lipid bilayer. Here's the extracellular part. Here are those tails. Here's just a bit of a close-up of that phospholipid. So it's actually got a bunch of phosphate, which is like a phosphorus surrounded by oxygen molecules, and some glycerol, which makes it hydrophilic, which means it loves water. You might know that, my chemistry teacher told me this, that polar molecules have FON, or a FON. So they love fluorine, I think, oxygen, and nitrogen. And you can see this has lots of oxygen molecules in it, which means it's more likely to like water. Well, hydrogen bonding is FON, I think she said. So hydrogen bonding, it loves hydrogens, loves water. FON, as well as some hydrogens, I guess, but mostly fluorines, oxygens, and nitrogens love water. So you can see just by having this many oxygen molecules means that it's really loving water. Here are the tails, which are hydrophobic tails. These are saturated fatty acids. So because the tails are literally fatty acids, they love lipids or fats. They are lipophilic, which means they must be hydrophobic. I know I'm using these words and you guys might not have come across them before. That's okay. Just try and take some time later to think about what hydrophobic, hydrophilic, lipophilic means. You'll notice here that there's also a kink in the tail, and this is due to an unsaturated fatty acid here. What this kink does is it kind of controls the fluidity of the membrane. So if lots of our little phospholipids have lots of kinks in their tails, this means that the membrane will be more fluid, because it means that instead of being super close to each other, that little kink in the structure pushes them apart a bit more, which means more fluid can go in. So I can kind of control the fluidity of the membrane. Okay, we've also got cholesterol. So they're usually drawn as like your little yellow hexagons. So there's little yellow hexagons all over. These are all cholesterols, and they can also interfere with the fluidity of the membrane. So they can actually control if the molecule sort of solidifies or not. So you can see he's like pushing her away. He's like keeping the side of the membrane apart. So it prevents the phospholipids from aggregating and solidifying, thus maintaining the fluidity of the membrane. So you can think of things like butter, for example. Butter is a solid, but perhaps if there were more cholesterols between the tails, it might be more fluid. You don't really have to know much more than cholesterol, increases fluidity of the membrane. It's very much what you need to know. As in, like, for that point. For this point here, it prevents the phospholipids from separating, kind of acts as a glue component to maintain the integrity of the membrane as well. So it just keeps those phospholipids there so that they don't drift away too much. Cool, so that was all we need to know about cholesterol. Hopefully you guys have a bit of an understanding about the phospholipids now. So now we're going to move on to transport across membranes. Just give it a time. Okay, cool. We're nearly like half an hour through. Okay, so transport across membranes. So we've got this word diffusion, which I mentioned a few times. I know I'd heard of diffusion in like this perfume would diffuse across the room, but I didn't really understand what it meant. And like you read the textbook and there were these big convoluted definitions. The passive net movement of molecules from a region of high concentration to low concentration. Like, what does that mean? It's just such a big definition. So I really encourage you guys to kind of break these definitions apart and kind of simplify them. So, passive. What does passive mean? Passive means no energy is used. So, the no energy, what's net? Net means overall. So, the low energy or no energy overall movement of molecules from a region of high concentration to a region of low concentration. What does that mean? So you'll see in this little GIF, I'm not really sure how you say that all of these molecules start close together. It's like when you spray a perfume, they all start, like come out the nozzle really close to each other, and then they spread out around the room and try and create like an even distribution across the entire room. Okay, so the passive net movement of molecules from a region of high concentration to low concentration. This means that the molecules enter the space, they move without using energy, and they move overall to create a very even distribution, whereby those molecules move from high concentration or very much in one area to spread over the entire area. Okay, so hopefully that makes a bit of sense. You can see it's kind of spread all over. It's like when you enter a house, right, and you've got the heater in the corner, and the heater's really warm, and it's really hot right next to the heater. But over time, that heat will spread to the rest of the room, okay? That's kind of like what diffusion is. Or if you pour some water into a glass. This one's a bit harder to kind of visualize because I guess it happens very fast. But when you pour water into the glass, it sort of like conglomerates in one area initially, and then it spreads all over. It's kind of like diffusing through the space, sort of. So those molecules are just moving to fill that space and create an even distribution. So this definition of diffusion can often be extended to say something like the passive net movement of molecules from a region of high concentration to a region of low concentration in order to establish an equilibrium. So you might see that definition written somewhere. So you can see we've got a region of high concentration here and a region of low concentration there. Those molecules are spreading from the high concentration areas to the low concentration areas in order to establish an equilibrium, or kind of spread very evenly throughout the entire space. So we've got quite a few definitions for diffusion, so we're kind of going to build upon that. So we've got that definition at the top, and we've got simple diffusion and facilitated diffusion, okay? So simple diffusion is just basic diffusion, whereas facilitated diffusion is kind of like helped diffusion. So facilitated is like to be helped. So what is simple diffusion? This is when things move directly through the bilayer. So they move through that lipid bilayer we talked about. So cells can like have alcohol, small substances like steroids. They can move through the bilayer because these are hydrophobic substances. Other very, very small uncharged molecules such as water, oxygen, carbon dioxide can also move directly through that lipid bilayer. So we go back here. We mentioned that hydrophobic molecules can move through the bilayer, right? Hydrophobic molecules. So, molecules which are scared of water. Which area of the bilayer is actually hydrophobic? Do you guys remember? So we talked about the phospholipids. They've got hydrophobic tails. So this entire part of the bilayer, this like little yellow taily bit, all along, that's hydrophobic, okay? So small molecules like water, alcohol, uncharged molecules, they can kind of pass through. If they're non-polar or hydrophobic, they would actually pass through quite readily because they've actually got to traverse this entire hydrophobic area. And you can kind of see that this hydrophobic area takes up more space than that hydrophilic head area, which is quite small and thin. So you can just like skim through very easily, okay? On the other hand, we've got facilitated diffusion because we actually need some hydrophilic molecules to enter our cells. And they might actually be too big or too water-loving to actually pass through, but they are quite essential. So we needed to figure out some way to like get them through, or our bodies did anyway. And so they came up with facilitated diffusion. So this is the passive net movement of molecules across the plasma membrane via transport proteins. And so this includes things such as large hydrophilic molecules, such as glucose or amino acids, and charged molecules such as sodium and calcium. So what this means is that they move along the concentration gradient. So say there's a ton of calcium here and it needs to get into the cell. Because there's so much there, your body likes to equalize stuff, so it's trying to make an equal amount on both sides of the membrane. So your body or the cells are going to try and get an equal amount of calcium on both sides of the membrane. So they're just going to pass through. And they pass through this channel protein or protein channel. So it's kind of like a little tunnel whereby things which are perhaps hydrophilic can pass through. Because if it's water loving, this little fatty tail area will hate it. They don't want to hang out. They don't want to be near each other. But if it's hydrophilic and it needs to get in, it can diffuse down its concentration gradient via facilitated diffusion and actually enter the cell by entering one of these little channel proteins. Okay? Okay, moving on from that, we also have active transport. And so this is transport which requires the use of ATP energy. So active movement of molecules across the plasma membrane against a concentration gradient, which sounds a bit counterintuitive. So let's have a look at this diagram here, or this GIF, I don't know how you say that word. So say this is like calcium. We see there's a ton of calcium already inside. So it seems counterintuitive to actually have more calcium pumped in, okay? But calcium or sodium or potassium is actually really important for our cells, and we actually need quite a lot of it inside the cells. So even if there's already a ton inside, our bodies or our cells are like, no, we need more. Let's bring all of it in. But it goes against the concentration gradient. The concentration gradient would be like usually, oh, exit. Let's balance it on both sides. But we're actually moving against the concentration gradient. And for this, we actually need to use energy. Okay? It's like if you were to roll something down a hill, like you go up to the top of the hill and you drop something and it will roll down pretty easily, right? But if you wanted something to go up the hill, it's a bit harder. You need to actually push it up the hill and use the energy to push it up. So that requires energy, or ATP. Okay, so that's what active transport is. It's like the movement of things across the plasma membrane against a concentration gradient. So you actually have some more channel proteins and carrier proteins, which can do that. So say you've got this channel protein, but instead of moving stuff down the concentration gradient, you're actually moving it against the concentration gradient. So say you've got a ton in here and barely any out here, you're still pumping that in anyway. And you've also got something called a carrier protein. Hopefully you've got a nice picture of that. I do not. But it's kind of like a mechanism which snaps open and shuts. So carrier proteins, that little molecule enters, and then once it enters, it snaps shuts it in. Okay? So protein channels and protein carriers, or carrier proteins, channel proteins. They're really useful. I'm just gonna get a tissue, guys. I feel like I've got hay fever right now. Good. Okay. Onto a different type of diffusion, osmosis. So you guys may have heard of osmosis. I think it's like a surfy type brand, like surfing. I don't really know, but that kind of makes sense to me because osmosis is the movement of water molecules. Okay, so movement of water molecules. So it's the movement of water molecules across a semi-permeable membrane from a region of high water concentration to a region of low water concentration. Just gonna get a drink real quick, guys. So the net movement of water from a region of, or across a semi-permeable membrane from a region of high water concentration to a region of low water concentration. Okay, so this is like a glass of water with a selectively permeable membrane in the middle. So water glass with a membrane in the middle. You can pour lots of water into one side of the membrane, and typically the water molecules would just move to balance out. Okay, that's not what's going on here. So just typically the water would try and balance out. Okay. What happens though here is you've got lots of sugar on one side. Okay. So on the left we've got no sugar. On the right we've got quite a lot of sugar. Okay, so there's no sugar on the left, lots of sugar on the right. Okay. So what would happen here is that you can't move the sugar across the membrane. That sugar is too big to cross that membrane. So what happens is the water crosses the membrane to try and balance out the sugar-water concentration. Okay. So the water, so even though the membrane's got the same amount of water on both sides, this water will actually move from this side of the membrane into this side of the membrane to try and establish an equilibrium. So that water will actually move into this right-hand side to try and create like an even water-sugar ratio, or try and have more fluid for more solid. So there's more solid here, so we need more fluid to balance it out. Okay, that's in very basic terms. So hopefully that makes a bit of sense. You will spend quite a lot of time on this in class. It is something which is quite fundamental to bio, so you will look at it quite a bit. But I think for now, we don't need to go into too much more detail about that. I guess this goes into a little bit more detail. We can do this. I just feel like I'm wary of like throwing so many new words at you guys. There's quite a lot to learn in bio, and we're kind of covering kind of foundation for like a whole year almost in this one lecture, although there is quite a bit we're not doing. But it is quite a lot of info, so please, if you feel like you're getting overwhelmed by how many new words and phrases and definitions I'm giving you, that's completely okay, because I'm giving you a lot. Okay. Hypertonic, isotonic, and hypotonic. Hyper means a lot. Hypo means like not enough. Okay, so hyper, too much, I guess. Hypo, not enough. Something which is like hyperactive is too active, whereas hypoactive is not active enough. So you can see here there is a very hypertonic solution outside the cell. What this means is that there is a ton of like solute outside the cell. So this area outside the cell is filled with fluid with too much solute going on. Whereas inside the cell there is a very small amount of solute. What this means is that the water will actually rush out of the cell in order to fill up this area in order to try and balance out the water to like solute concentration, to solute to these solids. Isotonic means there's an even distribution, so there's an even amount of like solid inside, even amount of solid outside, or solute, which is like molecules, I guess. And so the water is kind of quite balanced on both sides. Okay. On this side, on the other hand, we can see there's a lot of solution, or solute, sorry, inside, and not enough outside, which means that, you know, there's a high water solute concentration here, so the water will rush in to try and balance out the internal environment here, okay? So hopefully that makes sense. If that doesn't make sense, please pop the questions into the chat. Hopefully you guys are still putting messages in the chat and asking questions and things, and I'll try and clarify that. But I know it's kind of a big jump, like we do this in med school even sometimes, a bit harder than this, but like a similar kind of topic, and it can be a bit confusing sometimes. So no stress at all if it is a bit confusing. Sorry guys, I think it's like a very floral day today and lots of pollen. Anyway. Moving on to cell structure and function. So you guys might have heard of, I don't know if you guys have heard of this before, but the total surface area and volume of a cell can actually dictate how it behaves, sort of, in a diffusion-wise sense anyway. So this small cell here, this very small one, I might get the laser pointer out again. So it's a very small cube. It's got a length of 1, so 1 by 1 by 1. Also, length of the square, length by, or volume of cube is 1 by 1 by 1. So its total surface area is 6. Okay, so it's got 6 sides, 1, 2, 3, 4, 5, 6. So 1 times 1 is 1, times 6 is 6. The volume is 1. So 1 times 1 by 1 is 1. So our surface area to volume ratio is 6 to 1. Okay, 6 surface area, 1 volume. It's a bit abstract right now, but it will make more sense in a minute. The second cube, it's got a length of 2. So total surface area is 24. So 2 times 2 is 4, times 6 is 24, with the volume being 8, okay? So 2 times 2 times 2 is 8. So you can actually see we've got a smaller ratio. So 24 to 8 is actually equivalent to 3 to 1. And as our cube gets bigger, once again, now we've got a bigger surface area and a bigger volume, but our ratio actually gets smaller. And so as we progressively get bigger and bigger and bigger and bigger, that surface area to volume ratio gets smaller. Why do we care? So the surface area is the site which actually facilitates diffusion, okay, or osmosis. So things pass through that surface area, or that plasma membrane, and enter the cell. if something has more surface area, it's more capable of diffusion. Whereas something which has a very large surface area is not very capable of diffusion, or not as much as this one. Okay, so diffusion is really important because it can help facilitate metabolism, it can exchange gases or waste products with the outer environment, and help create like a very optimal cellular environment. It helps brings in important nutrients or amino acids or gases or whatever, and like shunt out waste products or metabolites that we don't really need anymore. Okay, so having this very small surface area to volume ratio is actually quite conducive to a lot of cellular processes. Okay, which is why cells are so tiny. Because if they were really big, they wouldn't be very effective at diffusing and getting all these like little metabolites in, or nutrients or wastes or whatever. So that's why cells are very small. Okay, that's why cells are so tiny. Okay, cool. So if you've got any questions about that, pop them into the chat right now. Just drop something before. Cool. Hopefully you guys are putting messages into the chat, and we'll move on to this topic in a minute. Just checking the time. Okay, we're going on. Okay, next section. So eukaryotic cell cycle. So this is quite an intense looking diagram, but you guys are going to cover something called mitosis and meiosis as well, and it's pretty much the cell cycle. So we're going to be looking at like the cell's entire life cycle, like what happens to the cell as it progresses and grows up and becomes like a teenager and then an old person and dies potentially. So we start with what should we start with? This diagram always confuses me because I always like look at it from the wrong side and try and like make sense of it, and then I go, wait a minute, that's not right. So we actually start from here. So we have a cell. They rest. They kind of like live in this like little hibernation stage maybe, called the resting phase, which is also known as G0, okay? Or, yeah, G0. So the cell may leave the cycle to enter the resting phase, or it may not. Like a lot of cells do start off and rest for a long period of time before they actually enter the cell cycle. So we've got G1, and this stage is where the cell actually grows quite a bit, like it gets like physically larger. Then we've got, okay, let's start from the start. So we've got the cell. Firstly, it enters interphase, which is like preparation for the cell cycle, okay? So it may enter the resting phase, which a lot of cells do. They like leave the cell cycle, enter rest, and they may like leave afterward or not. But yeah, a lot of cells do enter rest. They go through interphase, which starts with G1 or gap 1, which is where the cell grows. Like it physically gets bigger. Then they enter the synthesis phase. So you've got gap 1, synthesis, and then gap 2, which is the 3 stages of interphase, plus G0, which is like one other stage, resting phase. So G1, cell growth, synthesis is where you've got DNA being synthesized, or the S phase. And so what happens is that the cell actually duplicates its DNA, and you've got something called the centrosome duplicating. So you can see the centrosome here. It actually like replicates itself. So centrosome replicates. So you've got centrioles, microtubules starting to like be apparent maybe. But yeah, the main thing here is cell duplicates DNA, so DNA is copied, and it has the centrosome duplicating. The next stage here is the gap 2 stage, where it actually gets even bigger. So you can see it gets a lot bigger. You've got nuclear membrane, chromatin, and nucleolus there. The cell is just like finalizing its growing before it enters the actual cell cycle. You might have heard of the phrase like PMAT before, P-M-A-T. If that helps you, that's really good. My like histology and developmental bio teacher, professor at uni would be like, think of a dog and tell the dog, don't pee on the mat. So PMAT, P-M-A-T. So the first stage of the cell cycle is prophase. And so this phase is where the chromatin actually condenses into chromosomes. So chromatin, which is inside the nucleus, it actually condenses into like chromosomes, which we can actually see. So people often look at like DNA under a microscope. If it's in the chromatin phase, it's a bit harder to see. Chromatin is like the harder to see phase, but chromosomes are actually quite solid structures. They're like very tightly packed, and you can actually see it. The nucleolus also disappears. So the main thing to take away from prophase is chromatin condenses into chromosomes. Next up, we've got prometaphase, which is kind of like an intermediate phase. A lot of people don't really talk about this stage, and they kind of divide these things into either prophase or metaphase. But during prometaphase, we've got the nuclear membrane around that cell breaking down. Sorry, around the nucleus breaking down. We've got kinetical microtubules being produced. What they do is they invade the nuclear space and attach to kinetochores. So pretty much you've got, I know I'm throwing all these random words at you, they're very big words, but you've got your cell. Inside the cell you've got your nucleus, which is packaged in a nuclear membrane. Inside that nuclear membrane we've got the chromatin, which is like a bundle of DNA condensing into these hard, tough structures, which are easier to visualize. Then you've got the nuclear membrane breaking down. You've got kinetical microtubules, so you've got these like long, spindly arms trying to form. And they start to form and they attach to the middle of those chromosomes. Okay? You've also got polar microtubules pushing against each other to move the centrosomes apart. So these are centrosomes, these things, which are where the microtubules kind of originate from. They get pushed to the opposite poles of the cell. That's the prometaphase. So really don't be surprised if you guys don't do prometaphase, because usually these traits are divided into either prophase or metaphase. And I think Vika doesn't really discuss prometaphase. What is metaphase? So during metaphase, all the chromosomes line up along the metaphase plate, which is like an imaginary like line through the middle of the cell. So those chromosomes like line up, and you've got the microtubules attached to them, coming from those little centrosomes at the end. So those spindly microtubules are attaching to those chromosomes, and what they do is during the anaphase stage they just yank them apart, because you might be aware that chromosomes are made of like two arms. So I'll just draw them. Chromosomes, two arms. Sorry, that's a very bad drawing. They're kind of like almost butterfly looking. So two arms. And what happens is those microtubules yank them apart, okay, during the anaphase stage. So let's get that pen back. So the microtubules attach to their kinetochores, which is like the middle of those chromosomes, and then they yank them apart. So this one's over here. This one's over here, and they're still attached. So chromosomes break at centromeres, and sister chromatids move to opposite ends of the cell. So those sister chromatids are the two arms. Sister chromatids move to opposite ends of the cell. What happens next is that you've got telophase, and so telophase is where the nuclear membrane reforms and nucleoli reappear, and the chromosomes kind of unwind from that condensed state back into chromatin, which is less condensed, so kind of a more soft state, I would say. What happens next is that you actually have got myosin, which is a protein, I think, and actin actually contracting to cleave the cell in two. So it actually like slices a cell in half, and that's cytokinesis. Cytokinesis is the production of two individual cells, which is really the breaking apart of those two conjoined cells. And then eventually you've got two daughter cells. So that's a process of the cell cycle. So just to recap, we've got the resting phase. We've got the interphase, which is like the preparation, where a cell might enter the resting phase. Then we've got gap 1, which is where the cell grows. Synthesis, where we've got the DNA replicating and the centrosomes duplicating. Then you've got gap 2, where the cell grows even more. And then you've got the actual mitosis, which is that stage of the cell replicating. So you've got prophase, whereby chromatin condenses. You've got nuclear membrane breaking down. You've got centrosomes producing microtubules. Then metaphase, those chromosomes line up down the middle of the cell, and you've got the microtubules attaching to them. Anaphase, where they snap apart and break their sister chromatids so that they go to opposite poles of the cell. Then you've got telophase, whereby new nuclear membrane, lack of condensing now, so they're more in their chromatin state. And then finally, cytokinesis, which is the division of the two cells. Cytokinesis. Divide. Okay? Or like cyto-moving, like cell moving. Two cells move separately. So that's the stage of the entire, that's the entire eukaryotic cell cycle pretty much. I know that's quite a lot. You probably will need to watch quite a few videos on this or draw it out in your books and things because it is quite a lot of information. But I'm really just trying to articulate that when our cells replicate, like we've got millions and millions, probably billions of cells even. We need, we replace our skin cells and stuff constantly. And this is what happens, in our cells. This is how they, replicate and make copies of themselves. Okay? So it's just a basic background. I know that was quite a lot. Please let me know what you thought of that in the chat, or if that sort of makes a little bit of sense. You will cover it a lot in school, so don't worry about that. You'll do lots of pracs. We can actually visualize this, hopefully. A lot of schools do an onion prac, where they look at skin of an onion and look at it under a microscope and evaluate it. So it's a really cool prac, but yeah, I know it's a bit much right now, but it will get better. Okay, we're about an hour in, nearly. And let me see how many slides I have. Okay, cool. We're doing pretty well for time, actually. And we've actually just covered all of this, so. DMAT, prophase, metaphase, anaphase, telophase, and don't forget cytokinesis. I think I actually forgot to talk about cytokinesis one time on an exam, and I lost 1 mark. This was a practice exam, and I gotten everything else right, and it just got so bad that I forgot cytokinesis. It was really sad. So make sure you don't forget about cytokinesis. Cool. Any questions about that? Just pop them into the chat, you guys. Okay, so cytokinesis in animal cells. We have actually got this thing called a cleavage furrow, which is created by a ring of actin and myosin proteins. They create a ring around those two developing cells and cleave them in two. So they separate into 2 separate cells. They kind of pinch them apart. Okay? So we result in 2 separate daughter cells. In a plant cell, you probably won't do a huge amount on this in school, but just so you know, we've got vesicles being moved to the equator, where they fuse to form a tubular structure across the equator. So vesicles are just little storage facilities or storage boxes, and they all kind of migrate to the equator or the middle of that cell, I guess, and they form a cell plate. So they merge to form 2 plasma membranes of the 2 daughter cells, and this completes cell division. Pectin and other substances, which are found in plants, are deposited between the 2 membranes by exocytosis, forming the middle lamella, and cellulose is brought to the middle lamella by exocytosis, and cell walls are formed adjacent. I don't want to say don't learn this, but I wouldn't spend too much time on it. You don't really come across this very much. You don't look at this in 3-4 bio, which isn't to say you shouldn't learn it, but it's just something that doesn't come up very much. You might need to know a bit of it, but I wouldn't memorize it, is what I'm saying. Whereas this cleavage one is probably more important to you. Okay, so we talked about how bacteria or prokaryotes replicate via binary fission. And I was kind of deliberating over whether it's a more simplistic process. I think it's completely wild. It's literally cloning an entire thing. So bacteria often replicate like this. They reproduce via binary fission because they typically single cellular organisms. So you can see here we've got a plasmid. A plasmid is a ring of DNA. And you've got the ribosome, and then you've got some genetic material that kind of scattered. There is no nucleus or nucleolus because it's prokaryotes don't have membrane-bound organelles. What happens there is the whole cell is kind of copied. So you can see there's a copy being made. So that initial genetic material is copied, that plasmid ring is copied, and they kind of gravitate to opposite poles of that cell. So they move to opposite sides, and then you've actually got the formation of a split happening, and that actually causes a split there, and you've got 2 distinct cells being formed. That's binary fission. It's literally cloning the entire cell, and not just the cell, but often the entire organism, which I think is completely wild. But yeah, very interesting. Something to note is that you also get checkpoints in the eukaryotic cell cycle. So you've got G1 is a checkpoint. So in that interphase stage, we've got a checkpoint at G1. We know G1 is where we've got lots of growth occurring. So what we do with this checkpoint is that we actually check for nutrients, growth factors, and DNA damage. If there is some DNA damage, we might send the cell straight into resting state and try and rectify that, or just perhaps kill the cell. We also check for nutrients, growth factors, and DNA damage. I already said that, but yeah, just looking for those problems. The next stage is synthesis. That's fine. But G2, at the end of G2, we actually have another checkpoint, whereby we check for the cell size and DNA replication. Because DNA replication is a very precarious process, we want to make sure it's correct. Any errors in it can actually result in things such as cancer. We actually have a very, very important protein called P53. So P53 is like, we call it the guardian of the genome pretty much. It's a very, very important protein. It's a checkpoint inhibitor. What it does is it checks for mistakes in DNA replication, and if there is a mistake, which could be quite detrimental, it will like pause that cell cycle and prevent the cell from progressing further, because otherwise you may actually result in cancer. And so, like, you go out in the sun, you accumulate DNA damage. P53 will help to solve a lot of this. So without the P53, people are often more prone to cancers and things like that. Finally, we've got the metaphase checkpoint, which does, but hopefully does really well, checks for chromosome spindle attachment to make sure that those chromosomes are correctly attached, or attached to their microtubules. If not, you could actually result in conditions during meiosis, such as Down syndrome, or Turner syndrome, perhaps other like chromosomal level conditions such as that. Okay. I'm gonna drink. So I mentioned before that there could be DNA damage. What happens then? We don't want to keep a cell which is so damaged that it could actually like harm us and cause cancer. So what we do is we actually kill it, and we have something called controlled cell death, and this is called apoptosis. Apoptosis sounds like such a murderous word, right? Apoptosis. But that's what it is. It's controlled cell suicide, okay? Essentially, controlled cell suicide. So you've got two pathways. The intrinsic pathway, which is the mitochondrial pathway. So cell damage, including radiation, DNA damage, etc. It's kind of like recognized, and I think this pathway is kind of initiated from the mitochondria because it produces cytochromes. So this is a very complex diagram. You guys aren't going to do anything like this in high school, but once you get to uni, you might actually examine it in more detail. What happens is that the mitochondria recognizes maybe the damage. It sends out this thing called a cytochrome, cytochrome C in particular, and it creates a cascade of caspases, which are like enzymes. They all kind of conglomerate together to create this apoptosome and results in death. That's quite a full-on discussion of it. You don't need to know that much detail. What you do need to know is there is an intrinsic and extrinsic pathway of apoptosis. Intrinsic results in cytochrome C being released by the mitochondria, and this initiates apoptosis, or controlled cell suicide. The extrinsic pathway is also known as the death receptor pathway, which is such an intense name, I think. What happens is extracellular causes such as change conditions in extracellular fluid, infection, result in a signal being sent out, stimulating this like death receptor, and it also initiates apoptosis. So you really don't need to know too much detail, just knowing that there are two pathways, intrinsic and extrinsic, and intrinsic is the mitochondrial one. Extrinsic has the death receptor like binding and initiating this apoptosis. Something you also want to note is that there are these caspases. So you guys might have heard of enzymes before. Enzymes are pretty much any kind of molecule which speeds up the reaction rate of something. They're kind of like little, I don't know what you call them. They just do lots of the hard work in your cells, okay? They're like the workers. So caspases, anything ending in ase is an enzyme. So lactase, for instance. A lot of people are lactose intolerant, and you might take something called lactase. Lactase is an enzyme which helps to digest lactose. So caspases don't digest maybe, but they do work in the cells. Anything ending in ase is typically an enzyme, and they do a lot of the work, like digesting or breaking down or building or something like that, or sending signals. So this is the apoptosis pathway. This is something I would recommend understanding and being able to recount. My voice is dying. Okay, so apoptosis. So either intrinsic or extrinsic pathway signals, and what this does is it activates caspases. So you actually have caspase cascade. So a bunch of caspases activate other caspases, and they just go on a signal, but like turn on, guys. Look, there's a problem here. We gotta kill something. The final caspases break down the cell's cytoskeleton as well as proteins in the nuclear membrane and Golgi apparatus. So caspases send signals. Eventually, they break down the cytoskeleton of the cell and proteins in the nuclear membrane, as well as the Golgi apparatus. The cell will then shrink and form blebs. Blebs are like little bubbly bits. The cell breaks down into vesicles containing digested cell contents known as apoptotic bodies. Okay? So the cell breaks and forms blebs. Blebs, such a funny word. Little bubbly bits. And then you've got vesicles, which contain apoptotic bodies, which are like the poison to kill these cells. These apoptotic bodies are then released, so phagocytes are signaled to the site of apoptosis, and they phagocyze the apoptotic bodies to prevent inflammation. So the apoptotic bodies are the digestive contents. So I think I said they contain poison. The poison's already been released. They form apoptotic bodies, which are then digested by phagocytes. Because if you just have these apoptotic bodies floating around the cell or into other cells, they may actually incidentally destroy other cells as well, which don't need to be destroyed. So you want to control the suicide and make sure it's only killing the one cell which needs to be killed. Okay? So yeah, that's the apoptosis pathway. Caspase cascade, breaking down the cytoskeleton and internal organelles. The cell shrinks and blebs, which is then broken down to vesicles containing digested apoptotic cell bodies, or digested contents known as apoptotic bodies. Phagocytes are signaled the site of apoptosis, and they phagocytose the apoptotic bodies to prevent inflammation. Cool. So too much mitosis can actually lead to neoplasms. So this is when we've got too much mitosis. Too much cell division means we've got excess growth, bad growths that we don't need. Benign means they're kind of localized. They may not be that bad at the moment. Potentially malignant are localized, but they have the potential and will eventually invade other tissues and transform into cancer. And finally, malignant are the actual proper cancerous ones, which are quite harmful, and they can invade other tissues and cause lots of problems elsewhere in the body. So it's really important that we actually do regulate mitosis for this reason. If we don't regulate mitosis, if we don't have apoptosis and that controlled cell suicide, we may actually have too much growth, and that can lead to cancers forming. Okay? So I mentioned P53 before. P53 is that protein which is like the guardian of the genome, and what it does is it recognizes if a cell could become dangerous or could become a cancerous cell, and it kind of like suppresses that cell or the tumor. So P53 is a tumor suppressing gene. You've also got something called an oncogene. Oncogenes are more like, yes, let's keep growing. They have the potential to become cancerous cells. Or promote cancer production. So if anything goes wrong, we might have lack of tumor suppressing genes working, or too many oncogenes working. Too many oncogenes could mean uncontrolled cell growth and decreased apoptosis, induction of angiogenesis. What this means is that cancer cells need a blood source. They need nutrients which come in the blood, so they may actually have more vessels forming and giving them nutrients, and then invading and metastasizing elsewhere. Tumor suppressor genes are like the ones which prevent this. So if they are not working, or if they're a bit underactivated for any reason, then you could also have cancers forming. Okay? So there's a very precarious seesaw, I guess, of oncogenes and tumor suppressor genes, because we do need genes to signal, yes, build more cells. We need mitosis occurring, but too much of it can actually lead to cancers occurring. Okay? So it is quite a precarious line. Moving on from that, we're going to be looking at mutagens. So what are mutagens? They're pretty much anything which can cause a cell to kind of mutate and can potentially cause cancer. So things such as radiation. So UV radiation from the sun can actually cause mutations in your genome. So it can actually cause your genes to shuffle about, or nucleotides themselves to shuffle about, and can lead to cancers. So UV radiation from the sun, from X-rays, medical, dental, airport security screening, even from people get their nails done under a gel UV curing lamp, or even tanning beds. I think tanning beds are still legal in America, which I think is completely insane. really dangerous. So please don't go to any of them. Obviously, some of these things like X-rays are quite necessary for your health, but yeah, doing a lot of them constantly can be quite detrimental. And things like staying out in the sun without sunscreen. Like I feel like I did this in undergrad, and I did this every year in undergrad. But every time I did this unit, I just get so terrified, and I go out and buy like SPF 50 sunscreen and make sure I'm wearing it constantly and like reapplying and stuff because it's just kind of terrifying. Like you're really leaving so much down to chance. Like, what if P53 malfunctions and you're, I don't know, you gain too many cells and there's no apoptosis occurring, and it's just really scary to me. Other things like cigarette smoke, even just exposure to someone smoking, actually can induce like mutations in your DNA, which is also really deadly. So cigarette smoke just contains dozens of mutagenic chemicals. Nitrate and nitrate preservatives in hot dogs and other processed meats. I'm not really sure about hot dogs exactly, but I think salamis and things, and even not aspartame but other like fake sugars, they actually like deemed by the World Health Organization as carcinogenic because of the toxins and preservatives in them. So please don't eat too many of them. Barbecuing can create mutagenic chemicals in foods potentially as well, and even some chemicals like benzoyl peroxide or like acetone and stuff, I guess, quite harmful to your health. Other things such as infectious agents. So HPV is a sexually transmitted virus, or an STI, or causes STIs, and it's very deadly as well, which is why we get injections for it. But it can cause like cancers and things. Helicobacter pylori is another, like it's a food poisoning type agent that can also cause like harm to the cells. Which is why it's really important to just try and be healthy. So there are many environmental factors. So I think you do need to delineate between environmental factors and a few other things. But yeah, environmental ones are a very big one. Mutagens are factors which cause mutations in DNA, not mitogens. I'm not sure if we study mitogens in high school, but mitogens are things which cause cell growth, I'm pretty sure. Mutagens cause mutations in DNA. So carcinogens are anything which causes cancer. So a mutagen which affects TSGs, so tumor suppressor genes. That should be affects with an A. So a mutagen which affects tumor suppressor genes or proto-oncogenes include chemical factors, cigarette smoke, physical factors, ionizing radiation X-rays, and biological factors such as HPV and cervical cancer. So yeah, mutagens, carcinogens, these are really big environmental factors. Yeah. If you have any questions about that, pop that into the chat, you guys. We've got other factors as well, which can also affect your cells and can lead to mutations and things. So loss of immunity is one. Immunocompromised individuals may be unable to detect and destroy neoplasms. So you know when we get COVID vaccines, like during lockdown, we had to get vaccines, right? And we talked about herd immunity, which is something we will explore quite a bit in bio. But some people are immunocompromised and they may not be able to get vaccines. They might be allergic to something in the vaccine, like how some people are allergic to Teflon or, I don't know, latex and stuff. They may be allergic to something in the vaccine like that. So they can't get these vaccines. Or they may be immunocompromised in another way, or pregnant or something like that. And so they may not be able to actually get these vaccines. These sort of people often aren't able to detect and destroy neoplasms like this. They may have an immune system which doesn't recognize too many cells conglomerating, or they don't have enough tumor suppressor genes being activated. Stuff like that can be quite detrimental, and it can lead to development of cancers and things. What happens then is that can lead to the progression of malignant tumors, which can then be cancers. But often these are quite easily eliminated in a healthy person. So it's really important that we do have herd immunity, and we try and like eradicate or reduce carcinogens and things like cigarette smokes or poisons and viruses, like sexually transmitted viruses or other viruses as well, just to prevent the occurrence of someone. Like someone could literally get cancer from these things or die from it. Okay, this is a really good slide. I'm just get some water because it's pretty hefty. So we've got many hallmarks of cancer. We typically talk about 6 of them. I actually have a student, and I kind of explain this as like a vampire. Cancer is like a vampire, okay? So these are our 6 hallmarks. Evading growth suppressors, activating invasion and metastasis, enabling replicative immortality, inducing angiogenesis, resisting cell death, and sustaining proliferative signaling. Let's start with this blue one down here. Enabling replicative immortality. So vampires are pretty immortal, right? They live forever. What this hallmark is saying is that these cancer cells, they can pretty much replicate forever. Like, people talk about living forever, and hypothetically, it is kind of possible, but whenever it kind of starts to happen organically, it literally results in cancer. So, yeah, cancer cells pretty much live forever, like a vampire. Inducing angiogenesis. So angiogenesis is the production of new vessels to like create a new blood supply to go to more areas. So increased angiogenesis means that these cancer cells get more blood and more nutrients, therefore they can grow even more, okay? So kind of like a vampire, they want more blood. So live forever, more blood. They resist cell death. They just don't die. You can like try and kill this vampire, but it won't die. So they resist cell death. Cancer cells are quite good at just like evading death. Sustained proliferative signaling. They just keep growing. Maybe vampires don't grow so much, but we can just pretend this vampire keeps growing and growing and growing. It just keeps getting bigger and bigger, okay? That's what cancer cells do. They just keep getting bigger and bigger. They can evade growth suppressors. So anything saying stop, don't grow, they just go, no, I'm going to keep growing, okay? And what they do is they activate invasion and metastasis. What this means is that they actually invade other areas of the body. They might like get into the lymphatic system and just drift off to your lungs or, I don't know, elsewhere in your body and metastasize or get bigger. So like this vampire is creating a whole vampire community within your body. So live forever, more blood, resist death, keep growing, evade things saying don't grow, and just invade and metastasize, okay? That's what cancers do. So I always think of it as like a vampire doing that. A cell that is not yet differentiated and has the potential to develop into different specialized cell types is a stem cell. So moving on from cancers, now we're looking at stem cells, which are like very, very cool, interesting cells. So yeah, what is a stem cell? It's something which has not yet differentiated and has the potential to develop into different specialized cell types. Sorry guys, if you have any questions about cancer, please pop them into chat and we can just address them quickly before we move on to this next area. Anyway, so stem cells, we've got fat cells, cartilage cells, heart cells, nerve cells, blood cells, muscle cells, etc. Stem cells are like the precursor. They have the ability to actually specialize and become any of these developed cell types. Okay, we've got quite a few definitions to discuss. So we've got totipotency, pluripotency, multipotency. So totipotent is totally potential, which is very bad English, but it has total potential to develop into any other cell type. So they're like the best, most amazing stem cell because they can become any other cell, okay? The next level down, I guess, or like a little bit lower, is pluripotent embryonic stem cell. So they are stem cells which have quite a lot of potential. They can either become endoderm cells, mesoderm, or ectoderm cells. So ectoderm, they usually become either skin or like neuronal type cells. Mesoderm are usually muscles and blood, and then endoderm are like organs like liver, lung, I think liver, pancreas, etc. Or they give rise to lines, which give rise to those. Next up is multipotent, which is like a more specific category, so even smaller. So yeah, adult bone marrow, skin, cord blood, deciduous teeth, all of these can actually be induced pluripotent stem cells. So I think in 2015, a Japanese scientist won the Nobel Prize, I'm pretty sure, for actually figuring out how to get adult bone marrow, skin, cord blood, deciduous teeth, all of these kind of cells back into like a pluripotent stage. And they're called IPSCs, induced pluripotent stem cells. I can't remember what the final I is, but something like that. But yeah, they figured out how to actually transform these like adult cells back into pluripotent stem cells. And so this is really useful because it can be quite useful for organ transplants, or often people will reject organ transplants from someone else, but they won't reject their own body cells. So if you can get cells from one person and transform it into red blood cells or heart muscle cells or skin cells, then you could potentially use those cells in order to help heal this person without any kind of adverse reaction where the body goes, this is not mine, and freaks out and tries to destroy it. Okay? So it's a very interesting, exciting area of science. Something I really encourage in bio is not just sticking to the textbook, and maybe like if you find something interesting, go off and explore it, like look up articles about it, or I don't know if you guys have heard of The Conversation, which is a really good like a news source sort of, but it's really scientific news. It's very, very interesting. I love it so much. But yeah, seeking out those articles and learning more about it will actually benefit you in the long term because it will help with your exam, but also it's just very interesting, and it's quite fascinating to see the way that our society is evolving in terms of the science tech field. Cool. Okay, so embryonic stem cells, these are typically totipotent or pluripotent. So some people think it's not right to be experimenting on embryonic stem cells because these can be obtained from IVF programs and can replicate indefinitely. But these are often cells which are not used for IVF, they're like discarded afterwards. So yeah, some people have problems with this. But these cells are life-changing. They could help benefit the future of science hugely, right? They've got the potential to develop into any other type of cell and can be used for organ transplants or helping to heal people who are suffering. So I think it's a really amazing area to study. Adult stem cells are multipotent or unipotent, and they function to repair and regenerate damaged and aged tissues. They can't replicate indefinitely, and they can't really just transform to any other literal cell type, unlike totipotent stem cells. Cool. Okay, moving on from stem cells now. So if you have any questions about stem cells, you guys, just pop them into the chat. Cool. Okay, levels of cellular organization. So moving on now from stem cells and cancer and like human cells and things, we're looking at plant systems. Just have some more water. Okay, plant systems. Okay, so we've got specialized cells, tissues, organ system, organism. So I'm trying to figure out this diagram. Sorry, guys. Oh, okay. So the organism, the bigger one, like the biggest picture is the plant, the overall plant. And the systems which make up the plant are the vascular bundles, which make up the vascular system, which gives us like the nutrients, the fluids and stuff all the way up into the plant. Then we've got organs, which make up those systems, so things such as roots, and the roots are made up of tissue, such as root tissue, and this root tissue can actually come back, and the smallest component of that could be the root hair cells, okay? So all of these like layers of cellular organization make up the whole being, the overall plant. So what are vascular plants? Vascular plants are those which have specialized tissues for conducting water, minerals, and photosynthetic products through the plant. So like we have a vascular system, it's like our blood. Our blood is a bunch of pipes around our body which transport oxygen and nutrients to our cells. Plants have a similar thing, they've got a vascular system. So these have specialized tissues for conducting water, minerals, and photosynthetic products through the plant. Some plants are non-vascular. They have simplified tissues that absorb water directly through osmosis through their cell walls. So you can see this diagram of a plant here. We've got the epidermis, we've got some pithy areas, and vascular bundles there in pink. So there's little bundles. We've got phloem and cortex. Cortex is like the outer shell, or the outer layer. So in vascular plants, transport occurs due to vascular bundles that move water, mineral ions, and sugars around the plant. These vascular bundles are made up of xylem and phloem. So these are really important to know for Unit 1-2. So xylem transports water and nutrients from roots to leaves. Okay, so xylem from roots to leaves is the water. Phloem transports sugars produced from photosynthesis from leaves to roots. It's the other way around. So xylem going up, phloem going down. So phloem flows down. That's how I remember it. Lignin provides strength and structure to the plant. So lignin provides the strength and structure there. So you can see it's kind of making a very definitive pipe. We've got the xylem. Remember, the xylem goes up and it brings the water to the leaves from the roots, whereas phloem goes down and it's transporting those sugars and things gleaned from photosynthesis down to the roots. So you can see here in this diagram, we've got xylem, water minerals going up. There are no end walls between cells, and it's pretty much one way only. The outer cells of this root or whatever it is not living. With phloem, we're kind of going down. I know it's got things going up as well. I guess it's two-way movement. I always think of it as going down there. Okay, but it is two-way movement. You've got organic molecules such as sugars produced in photosynthesis going down. You've got end wall plates, which is sieve plates. You have two-way movement, and the cells are living, but they do need support. Transpiration. So water is absorbed through the root hair cells. It's actually pulled up the roots into the stem using cohesion, and it kind of pulls the water up like a vacuum. So they cohere to each other. Cohere, maybe. And it kind of pulls them up. And that's the process of transpiration. Once that water reaches the leaves of the plant, the water can actually exit through the stomata, which is like an opening. If the guard cells are full of water, so the guard cells are surrounding them, they will open, letting water out. If the guard cells are flaccid or like dry, they won't. They'll just close so that no more water can leave the plant. So it really depends on the guard cells. The guard cells kind of regulate and go, look, too much water, I'm kind of like full, let's open up and release the water. If the guard cells are shrunken, they will be dry. They'll be like, no, we shouldn't lose any water. Let's close that stomata and save that water. Okay? So stomata are kind of like the portal where that water is either conserved or like released. Humidity. So transpiration decreases when there is a lot of water vapor in the air. This reduces the water concentration between the leaves and air, so less water molecules actually evaporate. So water vapor, if that's heavy in the air, this will reduce the water concentration between leaves, so less water will evaporate. Okay? If the temperature is quite high, we actually have transpiration increasing. This is because heat energy increases the rate of water evaporation. Wind can also affect transpiration because as wind or air currents increase, the rate of transpiration actually increases as well due to the increasing rate of water evaporation. And time of day. The rate of transpiration decreases at night because it's cooler and more humid. So really all of these things can affect transpiration rate and affect the amount of water concentration within the plant and evaporation as well. So please put in any questions about this in the chat. Yeah. I'm so thirsty now. Okay, so translocation. What is this? It's the movement of sucrose from the leaves to the rest of the plant. So we know that photosynthesis typically occurs in the greenness of the leaves. That's where the chloroplasts are in the plant cell. So chloroplasts is the site of photosynthesis. Once those sugars are made, so sucrose and things are made in the leaves, they then have to move to the rest of the plant. So you can see here we've got the xylem vessel, which transports water up, or transpirates water, I guess. The water can flow into the phloem. You've got the translocation of sucrose going down, and then water moves back into the xylem by osmosis, which is that diffusion of water. So as I mentioned before, phloem transports the products of photosynthesis sugar through translocation. So here at this number 1 on the right here, we've got loading sucrose into the phloem. So that sucrose is loaded through a companion cell into the phloem. You can see this little, so it's the leaf source, which is where it's made inside of the chloroplast, where the photosynthesis occurs. We've got the water flowing down the concentration gradient and into the phloem, which increases the water pressure. When water leaves the phloem, the water pressure decreases and moves down the concentration gradient. So once again, we've got that phrase concentration gradient. So it moves from a region of high concentration to a region of low concentration. It then enters the companion cell into the root cell sink, and the sucrose is actually unloaded from the from the phloem into the sink where it's acquired. Okay. I don't think you need to know like a huge amount of detail for this, but just having kind of a basic understanding, especially the roles of xylem and phloem, is really important. Okay, moving on from that into animal systems now, and lots to talk about enzymes, hopefully. We'll be looking at the digestive system. So we've got the mouth, which is where the food enters. It then enters the esophagus, goes down through the stomach, passes through past the pancreas and the liver, enters the large and small intestines, and then leaves out of the anus. But along the way, you've got lots of digestion occurring, and this requires the use of many different enzymes. So what is the purpose of the digestive system? It's to obtain nutrients from food by breaking it down. And digestion is the breakdown of food into a form that can be used by an organism for metabolism. So like chemical processes. Digestion can occur due to physical and chemical mechanisms. As mammals are heterotrophs, they require eating other organisms to obtain their nutrients, and these nutrients are obtained from the breakdown of organisms. So once again, this is like the cellular organization. So we are humans. Like the system, which helps us here, is the digestive system. Organs which make up the digestive system include the small intestine. A tissue which makes up the small intestine includes the small intestine tissue. And a specialized cell includes the villi, which are like little hair-like structures which help with absorption of nutrients. Okay, we're doing quite a bit here. I feel like it is, like for digestive system, we will touch on enzymes and things, I think, but doesn't seem to be doing a lot here. Just kind of a shame. I love enzymes. Anyway, that's okay. So what we're really doing here is just giving you kind of a taste of a bunch of different systems and how they are important in the body. So we might look at the kidneys for a bit, actually. And then just go through a few more systems and then go into homeostasis. And then hopefully the last 10-15 minutes we'll just spend time talking about exam tips, study tips, techniques, etc. If you've got any questions about like exams or uni or biomed or medicine, stuff like that, you can put them in the chat too and I'll reply to them. But yeah, we're going to spend a bit more time just on these systems and then move into homeostasis and then just do study tips and stuff toward the end. So the kidneys have very like convoluted kind of structure or very complex type structure. They're situated like just beneath your ribs. They have lots of renal pyramids, interlobular arteries. They've got many different arteries. I wonder if all the arteries are written down here. No. You've got the renal artery here. It then divides into the interlobar artery, I think, and then you've got, I forget the names of these, arcuate arteries, interlobular arteries. I don't think you need to know all of these arteries, but they do divide quite a bit. You've also got the renal pelvis here, which is where what will be urine will flow through and then enter the ureters eventually. You've got the cortex, which is like the hard outer shell, I guess, of the kidneys. Which doesn't seem to be written here. Interlobular veins, or interlobar veins. Nephron, this region. Really, we're just looking at the structure of a kidney here. I don't think we need to go into that much detail. But what happens is that the kidneys are a site of like mass filtration. There is a lot of filtration occurring here. So you've got afferent arteries enter this loop of Henle. Where's the loop of Henle? Not here. So afferent arteries enter the glomerulus, and this is a site of filtration. They then enter the efferent artery and then continue through. They enter the proximal convoluted artery. Where is loop of Henle? Loop of Henle should be around here somewhere. Nephron loop, I guess that's it. And then they enter the distal convoluted artery eventually, and then enter the collecting ducts. So what happens is that along the site we've got lots of filtration occurring, and so the kidneys will filter ions and nutrients that we need, even water and sugar, and kind of decide if we need them or not, and then keep them or excrete them. So each kidney is made up of subsections called nephrons. The blood enters the kidney from the aorta, which is part of the circulatory system. It enters the Bowman's capsule, which filters out blood, cells, and proteins. Only water and small molecules can pass through. So there's the Bowman's capsule with the glomerulus within contained. The solution with water and small molecules passes through tubing, whereby we've got salts, glucose, amino acids, and water being absorbed. And anything that's not reabsorbed is actually excreted in the urine. So there are several different stages. So filtration, reabsorption, secretion, and excretion. So anything which is not reabsorbed is typically excreted. So we've got this formula here. Excretion is equal to filtration, take away reabsorption, plus secretion. Okay? So anything secreted is excreted. Anything which is reabsorbed is not excreted. So there's the kidney and its role in like our cellular organization system. So nephrons make up the kidney tissue, which makes up the kidney, which makes up the excretory system, which makes up humans. I think we just get a few more of them. One more, and then just a few slides about homeostasis, and then so we've got 61 slides. So we're nearly there, you guys. Cool. Okay, so endocrine system. Just a very brief overview of the endocrine system. So glands are organs located around the body. Endocrine glands produce and store hormones for release into the bloodstream. They regulate how much of each hormone is released depending on the levels of hormones or other substances already present in the blood. Things such as stress, infection, and changes in fluid balance can affect hormone levels. So we've got secretory epithelial cells. So epithelial cells are like very small cells, I guess. I don't really know why we're discussing epithelial cells right now. So I feel like we're doing too much of a broad overview. But you guys will look at this in more detail. So yeah, epithelial cells can make up glandular tissue, which makes up glands, which makes up endocrine system, and which can become a component of us as human beings. So it's really just about recognizing the bigger picture is made of much smaller structures and understanding that cells really underlie all the processes that we as humans have to go through. Okay, just a little bit on homeostasis in the last like 20 minutes, I guess. And then I'll just spend some time going over study tips and stuff and what worked for me during high school and undergrad in medicine as well. So what is a stimulus? A stimulus is a change in the environment, either external or internal. We've got receptors, which transform environmental stimuli into electrical nerve impulses. Impulses are then transmitted via neurons to the central nervous system. And the signal is then transmitted by neurons to effectors. Sorry, guys. Effectors are organs which produce a response to a stimulus. So, for example, here, if a temperature is too high, our bodies will recognize this, and the hypothalamus will recognize it, and it will kind of talk to the body. The hypothalamus will say to the skin, look, let's start to sweat. I'm just way too hot right now. It will talk to the blood vessels and stimulate vasodilation to the skin. Okay? So this vasodilation means that blood vessels toward the skin get bigger, whereas blood vessels elsewhere, like going to the working muscles, might get a bit smaller. This means that more blood is being shunted to the skin. As a result, we can actually sweat, and this causes the evaporation of that plasma, the sweat, and helps to cool us down. Hypothalamus also talks to the cortex, which is another effector, and says, look, find a cool spot. So our brains go, wow, it's really hot. I'm so hot right now. Let me find somewhere shady or cool to sit down and rest in. Okay? Cool. So this is a strange meme. They say 70% of you is H2O, while the other 30% of me is garlic bread. So the perfect handle doesn't exist. We obviously love garlic bread. I love garlic bread. Do you love garlic bread? Hopefully you do, because garlic bread. Anyway, so stimulus. I might be just sitting in my room here, and I might smell my mother cooking garlic bread in the oven, and I go, wow, garlic bread, and I smell that. That's the stimulus. The receptor would be my chemoreceptors in my nose smelling that magic, right? My nose goes, wow, garlic bread, so good. The signals then pass to the brain via afferent neurons. And then the brain then sends message via efferent neurons. As a result, the response is, I go and eat the garlic bread because it's so good, right? So that's kind of like a stimulus response model in kind of like a fun way. But try and have a go at applying it to different things. Like if it's really cold, what do? You might feel cold. That's a stimulus. Receptor. So like thermoreceptors and stuff in your skin might go, wow, it's really cold. Signals pass to the brain. What would be the effector? So stimulus, receptor, effector. Start to move somewhere cold, and response is, move somewhere warm. You might put a jumper on, or you might go and sit next to the heater. Something like that. Okay? This is just about hormones. So endocrine hormones. Hormones are secreted into the blood. That's what the endocrine system is. It secretes into the blood. And we've got other methods of controlling homeostasis. So thermoregulation, we just discussed that one. Water regulation. What happens when you're thirsty, right? So you might be thirsty. So you might think, you might have low water, right? You might think, okay, I'm thirsty. So your body starts to think, thirsty. So you actually have the hypothalamus talking to the posterior pituitary to release vasopressin, I'm pretty sure. I think to go and stimulate your body to actually go and seek out some water and drink some water. So you might actually go and drink some water, or you might stop running or something if you're running, something like that. or your body might also regulate how much water is leaving the body, stuff like that. Blood glucose regulation, you might like be feeling a bit faint, so you go and get a snack or something sweet, something like that. So there are lots of different ways of like controlling different actions and things controlled by your body. So it's really about homeostasis. So homeostasis overall is a means of kind of maintaining an equilibrium, like an equilibrium between the internal and external state. So you don't get too hot, you don't get too cold. Your body kind of keeps you in a happy medium. That's what homeostasis is. However, it can kind of malfunction. So I know I'm doing a bit of a brief overview and we're not doing a lot of detail here, but that's okay. You might actually produce too much thyroxine in your thyroid, which is like a gland just here somewhere. It's quite soft. You can actually kind of feel it actually. It's quite soft and squishy. If you have got too much thyroxine being produced, you can actually have an enlarged thyroid, and this can actually like erupt sort of or build up as a sort of goiter. And so there are many different thyroid disorders which can actually result in this. Or even like iodine levels, too much iodine could potentially result in this. So just being really aware of this, like, I don't know how much nutrients and stuff you take in, but also your body would typically regulate this. But if it doesn't, then you can have these malfunctions. You can have like the goiter occurring and have thyroid problems. People with diabetes often don't produce enough insulin, or their cells of the pancreas are unable to produce insulin. As a result, they may actually need to seek insulin from an external source elsewhere. You see in the normal condition, you actually have the production of insulin, I guess, or like it's receptive to insulin, whereas in this type 1 diabetes, the insulin is not working the way it should. Or those cells are not working the way it should. It's a bit of a confusing diagram, to be honest, but overall, typically our body should produce insulin, and this is really important in regulating blood glucose levels. But in the case of diabetes, we don't have this normal production of insulin occurring. Not really sure what's going on here. Maybe more insulin? I'm not sure. But overall, if our bodies are not like regulating homeostasis in a normal way, this can result in quite detrimental things for us. Okay, so we actually have about 15 minutes now to actually discuss exam tips and study techniques and things. So if you guys have any questions, please pop them into the chat. Just get another drink. Okay, so story time, I guess. When I was doing bio, so I mentioned before that I jumped from bio, like year 9 bio, was it year 9? Yeah, straight into year 11 bio, and I felt like there was just a big jump, and I didn't really understand what was going on. But like I would study really hard before my SACs and just kind of like cram a bunch of stuff and just memorize it for my SACs, and I would forget it the next day, which is a very bad method of learning, and I do not recommend it at all. But like I just didn't know how to study for bio, and so this continued all through year 11 bio, and then year 12 bio, or 1, 2, and 3, and 4 bio. And then eventually, I actually went to an 800 lecture in September when I was in year 11. So this was the year 12 bio September series lecture. And I remember kind of freaking out because I was next to a kid next to me, and he was like, I've already done 30 practice exams. And I was like, I've done none. And then we did a Kahoot. If you guys do Kahoots, we did a big Kahoot on the board at like RMIT, because it was a big lecture theatre, and I was attending this bio lecture, and it was happening at RMIT, and it was like a huge theatre, and there was so many people there, like over 100, I think. There was so many. And we were doing a Kahoot, and I got so many wrong. Like I think I got like one of the worst scores in the Kahoot in the whole lecture theatre, and I was just like, I've been doing so well all year, but I haven't really understood any of the content, nor have I retained it. I've just been memorizing stuff, which is really terrible, right? And so I was like, I need to like study. I have like 2 months into the exam. I can do this. So I went home and I literally printed out the entire study design, and I laminated it, and I stuck it up on my wall, and I like got a whiteboard marker, and I tick off dot points. And so I kind of worked through the entire study design and went through PowerPoints that my teacher had made, and just tried to teach every part of the study design to my pets, like my dog or my cat, or even like the wall or a stuffed toy or something like that. I would just try and teach it out loud, because everyone says like to teach is to learn, right? Like teaching is a really effective way of learning, and it is. I find it really is, because it helps me articulate, areas of study I perhaps don't really understand as well, or things I need to know. So I would have my notes next to me, and I would just try and teach stuff, and then I would refer back to my notes to check stuff if I needed to, or see if I was forgetting anything. And then I would like look back at the study design, no notes, and just try and remember as much as I could and teach it to my pets. I'd just sit my dog down and be like, photosynthesis time, let's do photosynthesis. So I would do that quite a lot. And I found it really helpful, okay? So I would do that a lot. I would teach it to someone else. That's the first thing I recommend, teaching your content to someone else. Or even with your friends before like a SAC or something like that. Teaching the content, trying to make sure you fully understand it. Like working through it out loud. And I find that like if I'm just quietly studying, I often like my brain starts to like drift off, and I'm like just reading stuff without actually concentrating. So I find that speaking often helps me to like actually grapple with what I'm saying and try and make like sense of it. And ultimately, you do have to explain all these concepts to an examiner at the end of it. So yeah, you do have to understand it. You have to be able to explain it to someone else. Which is why like teaching it to your pets or someone is really useful. Yeah. Anyway, that kid at the A-Tonnets lecture completely terrified me. So I was like, I need to like step up. So what I did after that was I printed off a ton of exams. I actually have a coffee table that I'm doing here, like a coffee table, and it was completely covered in exams. I had 20 exams, and you can get them from your teacher. You guys probably don't need to worry too much about this now, but it is a good study habit to kind of get into the habit of. So I printed out all the exams, and I just laid them out on my table, and I started working through them. And I had like an exercise book, and I would just like write down like all the multiple choice. I'd be like, okay, 1, 2. I think the bio exam has got 20 multiple choice or 40. I forget how many. But it has a lot of multiple choice. So I would do all the multiple choice over breakfast, because you can just have your exam there and just eat your toast and like circle what you think. And then I would do the other ones like during lunch break or something like that. So all the long or extended answer or short answer questions, I would do all of them. And I would just write it out in this little notebook, like just a workbook. And then I would actually go back and correct, or I would circle any question that I was unsure of. Doesn't matter if I like got it right eventually, but if I was a bit unsure about it, I would circle it or highlight it. And then I would go and correct the entire exam with the answers, because the VCE exams always give you answers on the VCE website. And I would like evaluate them all and see which ones I got wrong, and I would circle or highlight all the ones I got wrong as well. And so now I've got a bunch of questions which I've circled or highlighted. And these are questions I was either unsure about or questions I got wrong. And so what I would do is I would actually put them onto the A-Tonnets forum. So I like made an account and I had like an anonymous name. And I was like so scared. I think I said this, but like everyone seemed so smart. And I was like, I am doing so badly here. I'm not studying how I should be. I don't know what to do. But I was just trying my best. I would, every time I got a question wrong, I put it onto the forums. And so at the start, I was getting like 40% on my practice exams. And so I was like spamming the forums, and I was like, oh my god, people are going to hate me. And I did have a few people tell me things like, show us your working first, or what was your answer first? And then they would explain it. So I'd like have to like try and figure it out myself first. But people would like answer, and if I didn't understand something, I'd just be like, I still don't get it. And I feel like saying that out loud in real life is a bit hard, but saying it anonymously is a bit easier, because these people don't know you. Like if they're mean to you, like firstly, they're not mean, but if you do feel judged by them, they don't even know you, and you're really trying to study, so more power to you, right? Anyway, I would spam the forums. Every time I got a question wrong, I would spam the forums, and then I would get their answer and write it down in my notebook, go and consult the textbook, or like online, or a Roller, or any other kind of like website which helped me out. And I'd make a note of it in my book, so I'd be like, okay, I didn't really understand what a phloem was, and then I'd write that question up on the forum, and then get my answer, write it in my book, consult the textbook, whatever. Make sure I fully understood what a phloem was, and then try and teach it to my dog or my cat and be like, okay, I'm going to tell you what a phloem is, or what phloem are, okay? And so I would just do that continuously. And so I started off getting like 40% of my exams, and then I would just keep like going through areas of study. If I was feeling like, look, I got like 70% of, maybe 80% of cellular respiration questions wrong, I would just go back and study that entire area. And what I did was I actually bought a summary book. I got a really good one called A+ Bio Notes, but there are lots of other ones, like A-Tonnets does one too. I just really enjoyed this green A+ Bio Notes one, and I would actually annotate it and add my own mnemonics or drawings, because I think of weird drawings and diagrams and stuff. So I would often do little mnemonics and scribble them in the corners. and just try and memorize it like that. So yeah, I would do lots of mnemonics and drawings and add to it if there's anything I feel like there was a mistake or I forgot about or I needed added to that book from my textbook. Because I think the textbook is great. It's just hefty, just really big. So I would like add to it and just keep revising that. And I would kind of read that every night actually. So I actually managed to read the entire summary notes book because isn't huge. It's quite a small book. And I just worked through the entire thing, trying to teach all of it to my pets. Look at the slides my teacher gave me. Like, try and remember as much as I could, teach it to my pets, do a practice exam, try and do like 1 or 2 exams every day. You guys are at the start of the year. You don't need to do this now. You're not even in year 12. Probably not yet, because you're doing 1-2 bio. So you don't need to do that now. But it's just a really good technique to kind of think about. So, yeah, I would spend a lot of time doing practice exams, writing up the answer in the back of my book, until I had this entire, like, answer section or like mistakes section, and I would just keep rereading the mistakes section, and I would annotate it when I needed to, or add to it, or draw pictures on mnemonics and things. So I'd keep reviewing the mistakes section as well. And eventually I just started improving my exams really well. I really think you do need to do quite a lot of exams in order to feel competent on a particular subject, and I found doing 15 plus was really useful. And just in my own experience, students who generally do above 15 exams, practice exams generally do really well, typically get like 40 pluses. I actually did 20 bio exams, and I don't think doing much more would have gotten me a higher score, so I got a 49 for bio. But yeah, it also really depends on like your active learning as well. So really making sure you go back and review your mistakes and try and explain it and learn from it is really important. So yeah, that's what I did. And eventually I did really good at bio, which I was really proud of because I'd gone from like knowing nothing in that little review lecture, the 800 one, to like doing really well, and then getting into like biomed, which I really wanted to, and then eventually getting to med now. So I feel like it's really helped me out studying in that way. And I find like I do have an iPad and I really enjoy reading my slides and stuff from the iPad, but I find that I have to hand write notes in like a workbook with a pen to help me like actually consolidate it. I don't know why. It just feels like I concentrate better handwriting stuff in my book. But yeah, you guys, so that's what I did for my exams, and I found that really useful for most of my other exams as well. I just found it a really good way to study. So yeah, I think it's a really good way to study. I think it's really effective, and yeah, hopefully that helps you guys out. We still have like 5 minutes left, or 7 minutes, I think. So if you guys have any questions, please pop them in the chat. I'm sorry this isn't like a live lecture, because then I could just answer them now while you ask me, but yeah, just pop in the chat right now, and then we have 5, 6, 7 minutes left for me to answer them. And yeah, otherwise, you guys, good luck with your exams. Thank you for coming along, especially in the summer. I know it's kind of a drag to go and attend like a revision lecture or a pre-revision lecture. But yeah, thank you so much for coming along. I might just see if there's any other slides. But yeah, I definitely highly recommend having a look at atonnotes.com. Just like search it, like scroll the forums or scroll the actual like website, because there are lots of things there, and they've actually like redesigned the website. I think I mentioned this before, and it looks really nice. It's really sleek and nice to use now. Very easy to use. My internet's not actually working right now at the moment, so I have to like, I don't have any internet right now, but I would show you the forums if like once you finish this lecture, or even right now if you've got some time, maybe have a look on your laptop at the atonnotes website. Really useful, and just it's a nice area to study. It's like a nice study area. Like, you know, when you go to the library at school and all the kids are studying hard and it kind of motivates you to study like that, it kind of feels like that, but a virtual space, if you know what I mean. So yeah, I really enjoy it. Anyways, I don't think I have much more to add. Just if you've got any questions about bio or my other subjects or how to study for them, or any areas of study we covered in today's lecture that you might need a bit more clarification on, let me know. Just pop it into the chat and I can try and reply to you. Please make an account for atonnotes. You can even use like a spare email that doesn't have your real name or anything, or just a fake name or an anonymous name. I highly recommend it. Thank you so much for coming along, you guys. Definitely check out our other lectures if that's something that interests you. Yeah, cool. I think that's all I really have to say. So good luck with your studying. Have a really wonderful year. I hope you guys love bio. please reach out to me if you like, I don't know, send me an email if you end up doing bio, biomed, or med, or something like that, because I'd be interested to hear. And if my resources or study tips, I don't think I had many mnemonics in this one, but if I do the 3-4 lecture later on in the year, I'll have quite a few. So yeah, come along, please, and yeah, I'll see you guys later. Thank you so much.